Hello, I am Geneva Mea Casido. For today's video, we will explore two powerful natural enemies of insect pests, which are Serratia entomophila and fungi. These biological control agents offer environmentally friendly alternatives to chemical pesticides and are making waves in sustainable pest management. Let's dive into how this natural organism works to control pests, protect ecosystems, and improve agricultural practices. So now, let's discuss what is Serratia entomophila. Serratia entomophila is a newly discovered bacterium that has shown promise as a natural pathogen of the grass grub or Costylitra zealandica, a pest that significantly damages pastures in New Zealand. The bacterium causes a condition known as amber disease, which results in the larvae of the grass grub changing color to amber and eventually dying. As Entomophila adheres to the chitinous lining of the grub's foregut, where it proliferates, disrupting normal physiological functions. Its natural occurrence and ability to manage pest populations have led to its development as a biological control agent. Characteristics of Serratia entomophila. Serratia entomophila is a rod-shaped, gram-negative bacterium which is non-spore forming. It can be cultured easily in laboratory settings, allowing for mass production with high cell densities up to 4 times 10 to the 10th cells per milliliter. Unlike many other biocontrol agents, this bacterium does not form spores, making its life cycle unique among microbial agents used for pest control. So now let's discuss its parts and function. So the adhesion. The bacterium specifically adheres to the chitinous lining of the grass grub's foregut, a crucial step in infection. For growth, once attached, Serratia entomophila proliferates within the foregut, disrupting the grub's normal biological processes. Pathogenic mechanism. The infection causes the larvae to develop in amber color, a sign of disease progression, which ultimately leads to death. Serratia entomophila system classification. Its domain: bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Enterobacterales, family Enterobacteriaceae, genus Serratia, species Serratia entomophila. Its life stages or life cycle. On the environmental stage, as Entomophila is found in soil and pasture environments and can survive independently until it encounters its grass grub host. Host infection stage. The bacterium adheres to the foregut lining where it multiplies, causing disease symptoms. And lastly, its pathogenesis and host death. As the disease progresses, the grub exhibits amber coloration and eventually dies after the host's death. The bacterium can persist in the environment. So now, let's dig into the significance and importance of Serratia entomophila. Biological control. Serratia entomophila offers an environmentally friendly alternative to chemical pesticides for controlling grass grub populations. This reduces harm to non-target species and ecosystems. For mass production and commercialization. The bacterium can be easily mass-produced and applied in large quantities to treat affected areas. This makes it a cost-effective solution for pest control. For niche market success, while its use is primarily limited to New Zealand, as Entomophila exemplifies the potential for microbial products in niche markets where traditional pesticides are ineffective or prohibited. And lastly, sustainable pasture management. By controlling grass grub population, this bacterium helps to maintain healthier pastures, reducing the need for synthetic pesticides and contributing to sustainable agricultural practices. So now, let's discuss fungi. So what is fungi? Fungi are a diverse group of organisms that exist in various forms. Some decompose organic material as saprophytes, while others live as parasites on plants and animals. Their primary function in ecosystem is to break down complex organic matter, recycling nutrients and making them available for other organisms. They thrive in moist or wet environments, and because they can be easily cultured in laboratory settings, fungi are not only studied for their ecological roles but also for their practical applications in agriculture, medicine, and biotechnology. Some fungi are pathogens capable of infecting insects and causing large-scale outbreaks, epizootics that can devastate insect populations. These pathogens have become a significant focus for researchers exploring natural, sustainable alternatives to chemical pesticides. Characteristics of fungi. Cell wall. The fungal cell wall provides structural support and protection against environmental stressors. Its vegetative phase. Fungi exhibit either a unicellular form or as yeast, or multicellular form as mycelium. Hyphae. These are the filamentous structures that make up the mycelium, and they may be septate or divide by cell walls or non-septate. Reproduction. Fungi reproduce through both sexual and asexual means, producing structures such as spores, sporangia, and conidia for dissemination and infection. Habitat evolution. Fungi have evolved from aquatic environments, for example, Colomomyces, to terrestrial environments. Its example is Metarhizium, expanding their ecological niche. Parts and functions of fungi. Thallus. It is the vegetative body of a fungus that can be unicellular or yeast or multicellular or mycelium. Its hyphae. Filamentous structures responsible for nutrient absorption and spreading through the substrate. Mycelium. The extensive network of hyphae that forms the main structure of the fungus. Spores or conidia, it is the reproductive structures that allow for the spread and infection of new hosts. So fungi system classification. Fungi are classified into various subdivisions based on their evolutionary characteristic. One of these is the Mastigomycotina, aquatic fungi with motile zoospores, example Colomomyces. Deuteromycotina, terrestrial fungi that reproduce via non-motile conidia. Example is Metarhizium buiferia. So now let's go to the life stages and life cycle of fungi. The life cycle of an insect-infecting fungus begins with spore attachment. When the spore lands on an insect's cuticle, it starts to germinate, forming a germ tube that penetrates the insect's protective outer layer. Once inside, the fungus begins to infect the insect, spreading through its body and colonizing its blood and internal organs. This process typically leads to the insect's death within 7 to 10 days. Some fungi even release toxins to speed up these fatal processes. Then finally, the fungus produces new spores, either inside or outside the host, often in humid conditions, to reproduce and find new insects to infect, continuing the cycle. Fungi, significance and importance. Fungi play a crucial role in maintaining ecological balance by regulating insect populations. One way they do this is by causing epizootics or fungal outbreaks that help control pest species. This natural regulation supports healthier ecosystems. In pest management, fungi like Metarhizium Entomophila and bassiana are used as biocontrol agents. These fungi act as natural predators to pests such as locusts, mosquitoes, and agricultural insects. Metarhizium anisopliae is particularly effective against locusts and mosquitoes, while Beauveria bassiana is widely used in countries like Brazil and China for pest control in agriculture. However, there are challenges. Commercializing fungal biocontrol agents has been tricky due to inconsistent results in different environments and difficulties in mass production. So looking ahead, genetic engineering could improve fungal strains, making them more effective for pest control. This technology offers existing prospects, particularly in developing countries, where fungal biocontrol could be an affordable, eco-friendly alternative to chemical pesticides. This could be especially valuable in areas facing locust plagues or mosquito-borne disease like malaria, where traditional chemical methods are less effective or unavailable. So, in conclusion, while there are hurdles, the future of fungal biocontrol looks promising, offering a sustainable solution for pest management worldwide. In conclusion, both Serratia, Entomophila, and fungi represent natural biological alternatives to chemical pesticides in managing insect pests. Serratia entomophila is a bacterium that specifically targets grass grubs, offering a high degree of specificity and environmental safety, making it an ideal agent for managing pasture pests. Fungi like Metarhizium anisopliae and Beauveria bassiana, on the other hand, have a broader applications, affecting a variety of insect pests by infecting them through their cuticle. Both agents offer significant advantages in pest management, reducing the need for harmful chemical pesticides and promoting sustainability. However, challenges remain in terms of commercial success, environmental constraints, and large-scale application. Continued research in genetic enhancement, optimized delivery methods, and broader market acceptance is key to improving the effectiveness and accessibility of these biocontrol agents. Integrating these natural solutions into pest management programs can lead to more sustainable agriculture, healthier ecosystems, and reduce environmental impact. So that's all, and